Hello students. Today we discuss the next question, that is the explain the process of transcription in eukaryotes or the eukaryotic RNA polymerases. So जो introduction and requirements are the same uh, that we have already discussed about the transcription in prokaryotes. So in your notes there is the first the introduction part of the transcription, that is the transcription is a synthesis of mRNA from the DNA with the help of RNA polymerases enzyme. Then transcription is nothing but the synthesis of RNA molecules. Then this transcription also occur throughout the cell cycles during or depending the needs of the cells. Then this transcription involves the rewriting the genetic messages are coded in DNA to RNA molecules. Then these are very short life, being present only during the protein synthesis. Then the transcription is catalyzed by the enzyme RNA polymerase in both prokaryotes and eukaryotic cells. These फर्स्ट क्या होता है कि जो DNA segments will unwind before the transcription. Then these, after adding of the RNA polymerase enzyme, or they ultimately added some nucleotides. Then these RNA synthesis mostly takes place in 5' to 3' in directions, and the strands which is having the 3' to 5' of DNA from which RNA is transcribed, that strands is known as template strands or antisense strands, and the remaining 5' to 3' strands which known as the anti-template strands or sense strands. So first of all you write down this introduction same in prokaryotes and eukaryotes transcription. Then requirements, these I already discussed in prokaryotics. That is, first requirement is the promoters that are the present at the 5' end, which is also called as the upstream end, and these are the small sequence of the DNA which provides binding sites for the RNA polymerase. Then second requirement is the terminator. Terminators are the small sequence of DNA which provides the binding sites for the termination factors, and is present at the 3' end, and which is also called as the downstream end. Then about the structural genes, these structural genes that are the transcribed, and then fourth factor is the initiation factor. So in transcription the sigma is the initiation factor and the rho is the termination factor. Then the sixth requirement is the RNA polymerase. So in prokaryotes only RNA polymerase enzyme is required, but in case of the eukaryotic cells there are 3 different types of RNA polymerase are required in the transcription process. So these in RNA polymerase, first, second and third are required in the process of eukaryotes. Transcription. So first is the RNA polymerase first. RNA polymerase mostly it is found in the nucleolus, and these are responsible for the synthesis of ribosomal DNA. तो जब राइबोसोमल डीएनए राइबोसोमल आरएनए जब सिंथेसिस करने का रहता है तब वहाँ आरएनए पॉलीमरेज फर्स्ट एंजाइम यूज़ होता है, and it is precursors to the 3 ribosomal RNAs, that is 28S, 18S and 5.8S rRNA. जब rRNA प्रोड्यूस करते हैं तब वहाँ पे RNA पॉलीमरेज फर्स्ट वर्क करता है, एंड प्रिकर्सर मतलब इट इज द स्टार्टिंग पॉइंट. सो in case of the 3 different ribosomal RNA उसमें 20S है, 18S है एंड 5.8S rRNA में जो RNA पॉलीमरेज फर्स्ट है वो एज अ प्रिकर्सर यूज़ होता है. देन RNA polymerase second. This RNA polymerase second is present in the nucleoplasm and transcribe protein coding regions. Then it synthesizes precursor of mRNA and most of the small nuclear RNAs. The small nuclear RNA is required for processing of primer RNA. तो जो आरएनए पॉलीमरेज सेकेंड का रोल क्या मोस्टली कहाँ पे प्रेजेंट रहता है, तो न्यूक्लियोप्लाज्म में एंड प्रोटीन कोडिंग रीजन में, और जो मोस्टली न्यूक्लियोप्लाज्म में प्रेजेंट रहता है, और जो प्रोटीन सिंथेसिस में जो कोडिंग रीजन रहता है उससे वो ट्रांसक्राइब्स करता है. मोस्टली आरएनए पॉलीमरेज सेकेंड क्या सिंथेसाइज करता है, तो mRNA दैट इज मैसेंजर आरएनए एंड सम स्मॉल न्यूक्लियर आरएनए सिंथेसाइज करता है. देन थर्ड एंजाइम इज द आरएनए पॉलीमरेज थर्ड. सो इट also present in the nucleoplasm, and it synthesizes the varieties of small RNA including the tRNA and 5S ribosomal RNA. तो RNA पॉलीमरेज थर्ड है वो न्यूक्लियोप्लाज्म में प्रेजेंट रहता है, और स्मॉल RNAs एंड उनके साथ में जो ट्रांसफर RNA दैट इज़ द tRNA को सिंथेसाइज करने का काम है RNA पॉलीमरेज थर्ड करता है. सो there are 3 different promoters for the 3 different polymerases. तो ये तीनों जैसे भी वहाँ की कंडीशन रहेगी, मतलब जो आरएनए प्रोड्यूस होने वाला है उसके हिसाब से वहाँ पे आरएनए पॉलीमरेज ऐड होगा, उसके हिसाब से वहाँ पे प्रोमोटर ऐड होगा, तभी वहाँ का ट्रांसक्रिप्शन प्रोसेस स्टार्ट होगा. यदि हमें mRNA चाहिए तब वहाँ कौन सा एंजाइम यूज़ होगा, तो RNA पॉलीमरेज सेकेंड. जब राइबोसोमल RNA चाहिए तब वहाँ RNA पॉलीमरेज फर्स्ट यूज़ होगा. यदि हमें tRNA चाहिए तो वहाँ पे RNA पॉलीमरेज थर्ड यूज़ होगा. यदि हमें स्मॉल न्यूक्लियर RNA या स्मॉल RNA चाहिए तब वहाँ पे RNA polymerase second या थर्ड यूज़ होता है. सो these are the 3 different enzymes which is present in eukaryotic cells. तो शॉर्ट नोट के लिए खाली आपको यूकैरियोटिक RNA पॉलीमरेज के ऊपर भी क्वेश्चंस आ सकते हैं. सो दिस इज द रिक्वायरमेंट. देन द प्रमोटर रीजन्स फॉर RNA polymerase second is composed of 3 regions: the initiation box, the Tata box, and variety of upstream elements. These promoter regions for RNA polymerase first has rich in guanine and cytosine. So these promoter regions of RNA polymerase third, which located within genes itself. So many transcription factors are involved in binding of eukaryotic RNA polymerase to DNA. तो जब भी आरएनए पॉलीमरेज ऐड होगा तभी ट्रांसक्रिप्शन प्रोसेस स्टार्ट होती है. तो यहाँ पे जो आरएनए पॉलीमरेज के कंडीशन के हिसाब से वहाँ का जो प्रोमोटर साइज है वो डिपेंड्स है. तो आरएनए पॉलीमरेज यदि फर्स्ट जॉइन होता है तो दैट रीजन विल बी रिच इन ग्वानिन एंड साइटोसिन. यदि आरएनए पॉलीमरेज थर्ड ऐड होता है तो वहाँ पे जो जीन्स है, जीन्स ऑलरेडी they are work for the transcription or they work as a promoters. So according to the enzymes, the promoters will be worked on this transcription process. So next is the actual steps involved in the transcription. So there are 3 steps. First one initiation, second elongation, and third is the termination. So what's happened in the initiation? In initiation stage, there is the RNA polymerase second released specific transcription factor to bring about the initiation of transcription. And after the formation of complete transcription, initiation complex transcription process begins. तो यहाँ पे जो पहला RNA polymerase second है that are bind with this RNase molecule and they forms the Tata box. After this Tata box is attached with these different promoter factors and this FIDT binds to this Tata box to form the initiation committed complex. वहाँ पे एक complex produce होता है and then actual the transcription process will be start. So first there is the addition of this Tata box to the DNA molecules. Then in the elongation process, the all other transcription factors complexes are left behind as the RNA polymerase moves forward and elongating the mRNA chain. तो जो elongation stage में क्या होता है कि जो भी transcription factors है, these transcription factors will be removed from that place and the RNA polymerase moves forward to forming or elongating the messenger RNA. So in this figure there is the 3' to 5' DNA strand. At 3' there is the promoter site and T is the terminator site. In that there is the one is the template strand. On this template strand there is the initiation of the mRNA will be start. Then there is the new form mRNA and after forming the new mRNA there is the addition of termination factor, that is the Rio terminator factor, and these newly synthesized mRNA will be released from this DNA. So the termination factor termination, this reverse fact used for terminates the transcription and it separates the RNA and transcript from the template strands. रियो फैक्टर क्या करता है, जो ट्रांसक्रिप्शन प्रोसेस है ट्रांसक्रिप्शन प्रोसेस स्टॉप करता है और जो प्री मैच्योर्ड मैसेंजर आरएनए उसे जो टेम्पलेट स्टैंड से वो सेपरेट करता है. सो दिस इज़ द ट्रांसक्रिप्शन इन यूकैरियोट्स. दिस इज only there is the difference in between the initiation stage in between the prokaryotes and eukaryotes. बाकी जो elongation और termination process है, this will be same in prokaryote and eukaryotic cells. So I hope all of you are understood about this transcription in eukaryotes. First of all, you have to write down introduction and requirements. Requirements somewhat similar, only there is the difference between the enzyme RNA polymerase. So in case of prokaryotes, only RNA polymerase enzyme is used, but in eukaryotes there are three different class of RNA polymerase are used according to the need of need of the RNA molecules. So RNA polymerase is for RNA polymerase first is used in case of synthesis of ribosomal RNA. RNA polymerase second is used in case of messenger RNA and small nuclear RNA, and RNA polymerase third is used in case of synthesis of tRNA and also some ribosomal RNAs. So this is the difference in between prokaryotes and eukaryotes. Then there at the promoter side different factors are used. These different promoter regions for RNA polymerase third that are combinedly called as the Tata box, that is TATA box. When these steps involved in transcription: initiation, elongation and termination. At the initiation side, RNA polymerase first is bind with this DNA molecule. Then in the elongation, these transcription factors starts synthesize the new mRNA. Then at the termination, after adding the termination factor, that is Rio factor, these prematured mRNA will be separate from the template DNA strands. So this is the whole transcription process in eukaryotes. So I hope all of you are understood about this transcription, that is first step in the protein synthesis in prokaryotes as well as eukaryotes. So now I stop here.